Hi everyone! Welcome to our presentation on the distribution and activation of matrix metalloproteinase 2 in skeletal muscle fibers.
The study focused on matrix metalloproteinases, or MMPs, as we will address them in our presentation. These are zinc-containing enzymes in plants and animals. They're involved in degradation, the remodeling of the ECM. There are 6 different categories of MMP2: collagenases, gelatinases, stromylysins, matrolysins, and other membrane-type MMPs. The study will focus on one category, gelatinases. One type of MMP is MMP2 or gelatinase A. Gelatinase A is made by skeletal muscle fibers, secreted by cardiomyocytes. Most of the total MMP2 in cardiomyocytes is found in the cytosol. Gelatinase A also has a wide range of substrates, which include gelatin and elastin. They're involved in the skeletal muscle differentiation, regeneration, and repair. The main purpose of this study is to better understand the location and the amount of MMP2 that is found in the skeletal muscle, and also what is required for the physiological activation. In the study, we determined proportions of MMP2 that is located intracellularly and extracellularly. We also observed MMP2's ability to diffuse across muscle fibers and observed if MMP2 degrades the substrate gelatin at normal levels of calcium.
In this study, the researchers first performed gelatin in gel zymography in order to detect gelatinolytic activity of MMP2 in human and rat muscle. Then, SDS-PAGE was compared with zymography under native conditions. The researchers also examined different calcium concentrations to determine whether MMP2 degrades its substrate at normal intracellular calcium concentrations. Western blotting was also used to detect the abundance of MMP2 in skeletal muscle samples. The researchers compared the amount of MMP2 in skeletal and cardiac muscle with known amounts of recombinant MMP2 pure protein. This allowed them to quantify the absolute amount of MMP2 in skeletal and cardiac muscle. Lastly, the researchers compared MMP2 levels in intact versus mechanically skinned muscle fibers. And this allowed them to find the ratios of intracellular to extracellular MMP2. The researchers obtained biopsy samples of human vastus lateralis muscle. To determine the amount of MMP2 localized on the inside of skeletal muscle fibers, segments of skinned and intact fibers were separated from human vastus lateralis and rat soleus muscles. After testing 4 different antibodies for MMP2, the AB92536 antibody was determined to be the best for identification of MMP2 because it accurately identified MMP2 in rat and human skeletal muscle. The researchers also used anti-GAPDH and anti-SERCA2A to detect sarcoplasmic reticulum calcium pumps. To determine subcellular distribution of MMP2 in human muscle fibers, the researchers performed a Western blot, which can be seen in the figure on the left. Anti-MMP2 and laminin, which is a protein marker for ECM, were used to probe for MMP2 in human and intact skin fibers. The dark bands for laminin are present in intact fibers, and there are no dark bands in skin fibers. The faint bands seen for skin fibers are not significant. These results make sense because the fiber skinning procedure removed all of the extracellular matrix, and thus laminin would not detect any ECM in skin muscle fibers. On the right-hand side, a Western blot of human vastus lateralis muscle is shown. The muscle was treated with APMA, which converts pro-MMP2 to active MMP2. The researchers completed a fractionation of whole homogenates of human vastus lateralis muscle into cytosolic, membrane, and cytoskeletal fractions. Muscles were probed for MMP2, SERCA2A, and GAPDH using the respective antibodies. GAPDH served as a loading control. There was a faint band for pro-MMP2 and a very dark band for active MMP2. Faint or no bands were seen for membrane and cytoskeletal fractions. The dark band for active MMP2 indicates that the majority of MMP2 in human muscle fibers is diffusible in the cytosol, and little MMP2 is associated with intracellular membranes or present in the cytoskeletal fraction. This figure shows the relative amount of MMP2 in intact and skinned muscle fibers from the three subjects. The values were not significantly different. However, the mean values allowed the researchers to determine the proportions of total MMP2 associated with the extracellular matrix and intracellular environment.
In groups with 500 micromolar calcium concentrations or larger, the white bands were seen and represent gelatinolytic activity of pro and active MMP2 in muscle. A similar pattern of activity was seen in all four experiments. The muscle, again, was treated with APMA to convert pro-MMP2 to its active form of MMP2. It was seen that in the non-APMA treated muscles, which is denoted by the minus sign, for all concentrations of calcium, there was no gelatinase activity. And this is because there were no white bands present. In the APMA treated homogenate, it was seen that gelatinase activity was not evident until 500 micromolar of calcium. The dark bands at about 20 kilodalton at the bottom were most likely products of proteolysis of myosin and other muscle proteins that were produced during the APMA treatment period in the 10 millimolar calcium. They were possibly activated by MMP2 or calpains or other proteases. To the left, we see an assay that examined whether or not MMP2 can proteolyze gelatin at normal intracellular calcium levels in the absence of SDS page. Zymography was performed under native conditions using recombinant MMP2. The gel unfortunately did not run well, but it can be seen that MMP2 was proteolytically active based on the degradation, which is seen in the white columns. We can see that activity increases as we increase the concentration of calcium from 0 micromolar to 10 millimolar of calcium. The highest activity was seen in the 10 millimolar calcium concentration. To the right, we can see the gelatinolytic activity of MMP2 plotted against calcium concentrations on a log scale. It can be observed that the activity progressively increases with higher concentrations, and 90% activity maximally at 10,000 millimolar.
The overall conclusions of the research study were the following. First, AB92536 antibody was determined to be the best for identification of MMP2. After testing 4 different antibodies, it was seen that this antibody accurately identified MMP2 in rat and human skeletal muscle. Secondly, the study showed that in rat and human skeletal muscle, 30% of total MMP2 is associated with the extracellular matrix, while 70% of the MMP2 is found in muscle fibers. Finally, MMP2 in muscle fibers is freely diffusible. This finding was discussed in a previous figure, which shows that fractionation of homogenates of skeletal muscle into cytosolic or diffusible and non-diffusible components, which were the membrane and cytoskeletal fractions.